Hello, people, and welcome to Med Novice, where we make tough medical stuff make sense. And the topic for today's discussion is inflammation. So, for the upcoming 2 to 3 lecture videos, we will mainly be discussing acute inflammation, and the main focus of today's video will be the mediators of inflammation, right? So, what exactly inflammation is? Inflammation is your body's immune response to basically some infection or any tissue necrosis going on somewhere in your body, right? And what we do in inflammation is we basically allow the inflammatory cells, you basically allow the inflammatory cells the plasma proteins and the fluid, right? You allow these three guys to exit the blood vessel. They exit the blood vessel and then they reach to the site where action is required within the interstitial space, right? Now, this was the basic overview of inflammation. Now, inflammation can be of two types. It can either be acute or it can be chronic, right? And the key inflammatory cell involved in acute inflammation is neutrophil, whereas the key inflammatory cell that is involved in chronic inflammation is your lymphocyte, right? So it is lymphocyte, right? Now we will be starting a basic discussion on acute inflammation. So what happens in acute inflammation is that your basic goal is to eliminate the pathogen and clear the necrotic debris, right? And it arises in response to infection. So let's start with acute inflammation. This arises in response to infection and to be more specific, more commonly bacterial infection, right? Or tissue necrosis. So acute inflammation is basically characterized by two things. The first one is edema, which is due to the fluid leaving the blood vessel and accumulating in the interstitium, right? And the second characteristic of acute inflammation is the key inflammatory cell, that is neutrophil, right? So acute inflammation is characterized by these two things. First one is edema and the second is the key inflammatory cell, that is neutrophil. And another important thing about acute inflammation is that it is an immediate response, right? It is an immediate response due to which what we see is that this response has a limited specificity, right? It has a limited specificity. Like it is unlike chronic inflammation where you have specific antibodies for the specific antigen, and rather what we say is that the acute inflammation is a generalized response, right? It is a generalized response. Now we will be coming to the main focus of today's lecture, and that is the mediators of inflammation. Mediators of inflammation, right? So the first mediator we will be discussing is TLR. TLR, also known as toll-like receptors, right? So these TLRs are basically present on the cells of innate immune system, right? They are present on the cells of innate immune system. And what are the cells of innate immune system? They are the macrophages and the dendritic cells. Dendritic cells. Right. So what these TLRs do basically is that they recognize something called PAMPs. They recognize PAMPs. PAMP is basically abbreviation for pathogen associated molecular pattern, right? For example, you have CD14, the TLR receptor present on macrophages, and it recognizes the lipopolysaccharide membrane present on gram-negative bacteria, right? So you have the CD14 receptor on macrophages that recognizes the lipopolysaccharide membrane present on gram-negative bacteria. Here the CD14 receptor is basically the toll-like receptor, and this lipopolysaccharide membrane, it acts as pathogen associated molecular pattern, right? Now as the TLR recognizes the PAMP, it lets the body know that invasion has occurred, right? And now its function is to elicit response. So what basically happens further is that this TLR activation results in upregulation of a molecule. Upregulation of a molecule, that is NF-kappa B, right? So this is the molecule NF-kappa B. It is upregulated when toll-like receptor is activated. And upregulation of this molecule results in switching on of the immune response genes, right? Activation of immune response genes, right? And as the immune response genes are activated, there is production of multiple immune mediators, right? So there will be production of multiple immune mediators, right? And these multiple immune mediators will further enhance the response, right? Enhance the inflammatory response. So this is how TLR activation helps us. Now one more important fact is that TLR is also present on the cells of adaptive immunity, right? The cells of adaptive immunity are lymphocytes, right? So TLR are also present on the cells of adaptive immunity, and hence they also mediate chronic inflammation. So this is one important point to know. Now we will be moving to the second mediator, and the second mediator is arachidonic acid and its metabolites, right? So let's summarize them under a heading arachidonic acid. Now arachidonic acid is released from the phospholipid cell membrane, and it is released by the help of phospholipase 2. It is released by the help of phospholipase A2, right? And this arachidonic acid, it can undergo two pathways. The first one is cyclooxygenase pathway, and the second one is your 5-lipoxygenase pathway, right? The 5-lipoxygenase pathway. So when the arachidonic acid undergoes cyclooxygenase pathway, it produces prostaglandins. And the three major prostaglandins are: the first one is PGI2, that is prostacyclin. The second one is PGD2, and the third one is PGE2, right? So what these three guys do, they basically mediate vasodilation. And they also increase the vascular permeability. Right? And another important fact about PGE2 is that it also mediates fever and pain. This is particularly for PGE2. Easy to remember, right? E, the same phonetic sound is coming in fever. So PGE2 mediates fever and it also mediates pain. And let's discuss another general note here that the two mediators for pain are PGE2 and bradykinin. Right. These are some important facts which you can remember as notes, generalized notes, right? And this will be useful throughout the pathology lecture series. So we know that PGE2 mediates fever and pain is also mediated by bradykinin, right? Now let's see what happens in the 5-lipooxygenase pathway. When arachidonic acid undergoes this 5-lipooxygenase pathway, then leukotrienes are produced. Leukotrienes are produced, right? And let's see some of those important leukotrienes which we should know. The first one is LTB4, right? What LTB4 does is that it attracts and activates neutrophils. So the leukotriene B4, it attracts and activates neutrophils. And here we will be seeing another important note which you need to remember is that activation of neutrophil is done by 4 things, right? It is done by 4 things. The first one is LTB4. The second is the complement protein C5a. The third one is the IL-8, interleukin 8, and the fourth one is bacterial products. So again, you can remember this as a generalized note. Wherever you will see LTB4, C5a, IL-8 or bacterial products involved, you will see that the neutrophils are attracted and activated, right? So this was another general note. So 3 general notes we have seen. And now moving further to other leukotrienes, this was LTB4. Apart from this, the important ones are LTC4, D4 and E4, right? And what these leukotrienes do, their function is vasoconstriction. Their another function is bronchospasm. And another important function is to increase the vascular permeability, right? So these are the functions of leukotriene C4, D4 and E4, right? Now this increase in vascular permeability basically happens by contraction of something called pericytes. This happens by contraction of pericytes. So let's say this is your blood vessel and these are your endothelial cells here. These are your endothelial cells here, right? And here also you have these endothelial cells. Let's avoid drawing nucleus for some time. And you have a cell called pericyte present here, right? You have these cells called pericyte present here. What happens is when they contract, right? When the pericyte contracts, it basically pulls these endothelial cells apart, right? It will pull these endothelial cells apart, resulting in creation of pores here, right? Due to which what we see is that the vascular permeability increases. Another thing that this leukotriene C4, D4 and E4, what they do is they induce smooth muscle contraction, right? They induce smooth muscle contraction, due to which you can see that bronchospasm also is one of the roles which is played by these leukotrienes, right? Now with this we conclude our discussion on the metabolites of arachidonic acid, which under the COX pathway, the cyclooxygenase pathway, was your prostaglandins, and which under the 5-lipoxygenase pathway was leukotrienes, right? And we saw the important ones and their functions. Now we will be moving to the third mediator of inflammation, and that is mast cell. We all know that mast cells are widely distributed in the connective tissue, right? They are widely distributed throughout connective tissue. Now what activates mast cell? So it is activated by three things mainly. The first one is tissue trauma. So mast cells are activated by tissue trauma. Apart from that, they can be activated by some of the complement proteins, mainly C3A and C5A, right? And the third way to activate the mast cell is by cross-linking of the IgE present on its surface due to the antigen, right? So cross-linking of IgE by antigen, right? So let's say you have a mast cell here, right? And you have one molecule of IgE here, one molecule of IgE here, right, on the surface of mast cell. And you have this antigen that is leading to the cross-linking of these two IgE, right? Leading to the cross-linking of these two IgE. This can result in activation of mast cell. So these are the three main methods by which mast cells are activated. And when they are activated, what basically happens is they undergo degranulation, right? Activation results in degranulation. So what happens is that when mast cells are activated, they can produce two types of response, right? The first one is the immediate response. The first one is immediate response, wherein what happens is that the mast cells have some preformed histamine granules, and as we know that mast cell activation leads to degranulation, then from those preformed histamine granules, histamine is released, right? Histamine is released. And this released histamine basically leads to vasodilation and increase in vascular permeability. Right. So this is the immediate response which we see on activation of mast cells, where histamine is released leading to vasodilation and increase in vascular permeability. Now mast cells also have a delayed response, right? They also have a delayed response. And what happens basically in this delayed response is that arachidonic acid metabolites are produced. Arachidonic acid metabolites are produced. They are also known as icosanoids because this arachidonic acid has 20 carbon atoms in it, right? So icos is the IUPAC term standing for 20. So hence they are also known as icosanoids, derived from a icosanoic acid, right? So what happens in this delayed response is that production of these arachidonic acid metabolites, particularly the leukotrienes, right? What happens is that the acute inflammatory response is maintained even after several hours. So the delayed response helps in maintaining the acute inflammatory response. The delayed response of mast cell helps in maintaining the acute inflammatory response, right, after several hours. So with this we conclude our discussion on mast cells. Now we will be moving further to the fourth mediator, and that is your complement system. The complement system. So complement system is basically consisting of a group of pro-inflammatory serum proteins that complement inflammation, right? So they are a group of pro-inflammatory serum proteins that complement inflammation, and they circulate as inactive precursors, right? Now as they are circulating as inactive precursors, what is required to execute their action? Activation, right? So activation can be by three pathways. Activation can be by three pathways. They are quite simple. The first one is your classical pathway. And what happens in the classical pathway is that you have a complement C1 that binds with the antigen bound IgG or IgM, right? IgM. And basically we know that the initial response to any pathogen is mediated by these two immunoglobulins. The first one is IgG and the another one is IgM, right? IgM is a pentameric form, so in that way it helps in responding to the major amount of pathogen present there. And IgG is the smallest immunoglobulin, so it is produced in quite high amounts, due to which again it helps in countering the pathogen in a better manner, right? So this is how classical pathway works. C1 binds with antigen that is bound to the IgG or IgM, and it leads to activation of complement cascade, not coagulation cascade, but the complement cascade, right? So this was the classical pathway. You have C1 binding to the antigen which is bound to IgG or IgM, which are responsible for initial response to any pathogen, leading to activation of complement cascade. Now moving to the second pathway. The second pathway is the alternate pathway. Alternate pathway, right? What happens here is that there is direct activation of the complement system by microbial products. So microbial products lead to direct activation of the complement system, right? And the third pathway is the MBL pathway, also known as the mannose binding lectin pathway, right? What happens here is that this mannose binding lectin, it basically binds to the mannose present on microorganism, right? So MBL binds to mannose present on the microorganism, right? So these were the three pathways which helped in activation of the complement system. Now the result of all these three pathways is generation of C3 convertase, right? So at the end of all these three pathways, you have something called C3 convertase that is being generated. C3 convertase generated. Now as the name is clearly indicating, C3 convertase would be converting C3 complement into C3A and C3B. Now if you remember, one function we discussed about C3A was that it led to activation of mast cells, right? And this other portion, that is C3B, what it will do is it will join and help in producing C5 convertase, right? Again, what will the C5 convertase do? It will convert C5 complement into C5A and C5B. Again, if you remember from our previous discussion in this lecture itself, C5A was discussed at two instances. The first one was that C5A attracted and activated neutrophils, right? The four molecules, the four things that basically attract and activate neutrophils, that is the leukotriene B4, C5A, IL-8 and bacterial products, right? So C5A will lead to attraction and activation of neutrophils, and it also activated mast cells, right? C3A and C5A were the two complements that led to activation of mast cells. So C5A and C3A functions we know. C3B also we have discussed. What this C5B does is that it complexes with complement C6 to C9, right? All these complements basically form a complex, and that complex is known as membrane attack complex. Membrane attack complex, right? The MAC. What this membrane attack complex does is that it produces hole in the plasma membrane of microorganism. And if you remember from the discussion of cell injury, the hallmark of irreversible cell injury was membrane damage, right? So now as the membrane has been damaged for the microorganism, what will ultimately happen is cell death, and there will be lysis of that microorganism. So this is how the complement system works, right? So once we will be discussing the key products of complement system. The key products were C3A and C5A. They trigger mast cell degranulation. Apart from that, C5A was a chemotactic factor for the neutrophils, and C3B was the opsonin for phagocytosis by neutrophils, right? C3B, and it also helped in the production of C5 convertase, right? And another key product of the complement system is the membrane attack complex that basically damages the membrane of microorganism, leading to the lysis of microorganism. So this was all about the complement system. Now we will be moving to the final mediator of our discussion, and that is the Hageman factor, right? Again, Hageman factor is something which is pro-inflammatory, right? So it is present in an inactive state, and it is activated when it is exposed. Whenever there is exposure to subendothelial or tissue collagen, right, then the Hageman factor will get activated. And after activation, what happens is it will activate some systems, and the main systems activated are the coagulation system, the fibrinolytic system, right? The involvement of Hageman factor basically in activation of fibrinolytic system helps this factor to play an important role in DIC, right? We will see what DIC is exactly further in discussions, right? And this is the reason why Hageman factor is playing an important role in DIC, and it also plays an important role in severe gram-negative sepsis. We will be discussing this in future lectures. So it activates the coagulation system, the fibrinolytic system. It also activates the complement system. And the Hageman factor activates the kinin system, right? So what happened basically in kinin system is that you have something called the HMWK, a high molecular weight kininogen, which is cleaved, leading to formation of bradykinin, right? And this bradykinin helps in release of histamine, and also it is a mediator of pain, we know, right? So what will be the entire function? It will be vasodilation, increase in vascular permeability, and mediation of pain, right? This is done by the activation of kinin system, and it is activated by Hageman factor. So this was all about the mediators of inflammation, and now we will be discussing the final topic for today's lecture video, that is the cardinal signs of inflammation. So the symphony of all these mediators results in cardinal signs of inflammation. And what are those cardinal signs of inflammation? Right. The first one is redness, also referred to as rubor, and warmth, also referred to as calor. So what happens is when the amount of blood flow increases due to vasodilation, it results in redness and warmth, right? And how is this vasodilation happening? It is happening by relaxation of arteriolar smooth muscle. Now, what are the key mediators for these two cardinal signs? The key mediators are histamine, bradykinin, prostaglandins. Right. So these are the key mediators for redness and warmth. Right. Now moving to the second cardinal sign, that is swelling, also referred to as tumor. Right. And this is due to leakage of fluid. Right. When the fluid leaks into the interstitial space and accumulates, it results in swelling, right? And what are the key mediators for swelling? The key mediators are histamine and tissue damage, right? Tissue damage resulting in disruption of endothelium, right? Tissue damage that disrupts endothelium. And what histamine does is it helps separate the endothelial cells, right? So this is the second cardinal sign, swelling, which is due to leakage of fluid, and the key mediators for swelling are histamine and tissue damage that would disrupt endothelium, right? Now moving to the third cardinal sign. The third cardinal sign is pain, also referred to as dolor, right? And we know that pain is mediated by bradykinin and PGE2, prostaglandin E2, right? And finally coming to the last cardinal sign of inflammation, that is fever. So what happens basically is that when you have pyrogens in your body, they act on the macrophage. They act on the macrophage, and the macrophage releases interleukin 1 and tumor necrosis factor, right? So IL-1 and TNF are released from the macrophage and they enter the bloodstream. Now what these two guys do, they basically increase the cyclooxygenase activity in perivascular cells of hypothalamus, right? We know that hypothalamus has a role in thermoregulation. So whenever a pyrogen enters the body, it activates the macrophage to release IL-1 and tumor necrosis factor, which would increase the cyclooxygenase activity in perivascular cells of hypothalamus, right? And what will happen when the cyclooxygenase activity increases? We know that prostaglandins are produced, and when the levels of prostaglandin E2 increase, then the temperature set point within the hypothalamus is increased or raised, right? Due to which what happens, the body temperature goes up. The hypothalamus regulates according to this new set point of temperature, which is higher than what it should be normally, right? Due to which the fever response occurs. So these were the cardinal signs of inflammation, and with this we conclude today's discussion on inflammation. We will be continuing on acute inflammation in the next lecture, and if this video helped you, don't forget to like, share and subscribe. This is Med Novice, learning medicine the simpler way. Thank you.